All right, we's back, and we're gonna be talking about meiosis. But I forgot to show you guys some videos, so I want to show you some cool videos first. And I think I've shown some of these already, but we're gonna show them again. Okay, let's see. Ribosomes, nope. Okay, I'm sorry, I'm in the wrong PowerPoint here. Oh, this is kind of cool, actually. I'll just show you this. This is a little chromosome here. A little electron micrograph of a chromosome. It's pretty nifty. Okay, let's see. All right, this is a good little video of a cell going through mitosis. So this is like from the last lecture. So that's the nucleus there. This is kind of the outside of the cell. And you can see we're in like prophase. All that DNA is packing up. Then the nuclear envelope breaks down, like right around there. Nope, yeah. And now it's going to be entering into metaphase. So the chromosomes are going to line up in the middle. See, they're not quite there yet. They're getting there. Got this kind of guy over here to the side. There we go. Okay, now he's all in the middle. Okay, perfect. Now we're fully in metaphase. We got these chromosomes being tugged back and forth. And then boom, anaphase, they split up. Bingo, we got telophase and cytokinesis kind of happening simultaneously. So you can see how like the DNA is kind of packed, but then it gets really tightly packed. And you can really see those chromosomes. It's really cool. Okay, that's one video I wanted to show. This is a little video I took I wanted to show. I think I've shown this before, but this is again in a frog embryo. It's really fast, really sped up. You can just see the DNA go from metaphase all the way to telophase and then cytokinesis, that actin coming in to pinch that cell into two. Really nifty stuff. Okay, shown those. This, no, don't want to show that one. This is a plant cell, but it's a really good one to see.
As this plant cell.
I don't want to listen to her. But you can see you can see the chromosome so good in this one. So that'd be like, kind of like late prophase, early prophase, and then late where the nuclear envelope breaks down. And then you can just really see those chromosomes so beautifully line up in the middle and then split up with anaphase. It's really cool.
First condense.
Okay. One last one I wanted to show, and I think this one does have audio, so I'm just going to shut up for it.
This visualization isolates a single chromosome at mitosis. The chromosome contains two identical copies of DNA. Each DNA molecule is packaged up into one of the sausage-shaped chromatids.
Sausage-shaped?
Sticking out from either side are microtubule fibers from the mitotic spindle, used for orienting and guiding the chromosomes to their correct positions. So
these are the microtubules attaching to the chromosomes. So this is one sister chromatid, this is the other sister chromatid. You see there's actually multiple microtubules that attach.
The red region is the kinetochore, which works as the interface between the microtubules and the chromosome. The kinetochore is one of the largest and most complex molecular mechanisms inside living cells, with multiple functions to perform. The kinetochore is central to the movement of the chromosomes and is able to hang on to the dynamic ends of the microtubules. It is also a mechanical tension sensing system, which is able to feel if everything is correctly attached and positioned. It is also a stop signal broadcasting system, sending out chemical signals to the rest of the cell about whether it is ready to undergo separation of the chromosomes. This kinetochore has sensed that the tension...
So this is a growing microtubule. It's polymerizing at this end. And all of these other proteins kind of make a ring around it. And later you're going to see this microtubule fall apart. So it's going to like open up like a straw that you cut and like peel back. And those parts that actually peel back push on that ring to move this whole thing forward. You'll see that.
It's just right, and the chromosome is correctly positioned and attached. The last little bit remaining red is the stop signal broadcasting system. It is carried away by a dynein motor, walking down the microtubule away from the kinetochore. These are molecular motors involved with a directed transport along the mitotic spindle. The orange proteins walking to the left are kinesins, molecular motors carrying proteins on their back. Striding from the kinetochore on long molecular legs are dynein, carrying the stop signal broadcasting system. When all the kinetochores are ready, the cell transitions into the anaphase stage, with the chromatids pulled apart to the opposite poles of the cell.
So this, oops, let's go back.
With the chromatids pulled apart to the
So that right there would be the centromere that's holding them together. And on this side, it's the kinetochore.
opposite poles of the cell.
We don't actually see that depolymerization, but that's okay. It's really kind of crazy and nifty. And like that is happening millions of times all throughout your body. Hundreds of millions of times, like every second. This is crazy. Okay. All right, that was all just basic cell division, mitosis stuff. Now let's get back into meiosis, which occurs right here in our crotchal region to make our gametes. So, meiosis has to do with, in females, making the egg, and in males, making the sperm. Okay, and we already talked about the basics of those kind of like sexual reproduction. Now we're going to say, how do we actually make this? Because we want to have the genetic material, cut it in half, so that when whoosh, they fuse, we now have the normal 2 copies of every chromosome, right? Let's go back to this picture just so it really fully sinks in and makes sense. Up here, right there. Like every human, we have 2 copies of 1, 2 copies of 2, 2 copies of 3, 2 copies of 4. So meiosis, it's essentially like we're getting rid of 1/2 of every one of those. Okay, we're making a haploid cell. So we're splitting off 1 from itself. Okay, let's get back in here. And this one's a little more complicated. There's extra little steps. But all of the interesting stuff, it actually has 2 phases. And I'm just going to say this right at the top. All of the interesting stuff happens in meiosis 1. Okay? Meiosis 2 is pretty much just mitosis. Okay, so all of the interesting stuff happens in meiosis 1. Okay, remember that. Okay, let's go through this and we'll kind of simplify it as much as possible. We're talking about how we make our gametes, our spermies and our eggies. All right, so we got a cell and it's got a nucleus. And we'll start in G1 just for good understanding. G1. Okay, we're going to draw an extra chromosome this time. So we're going to do a big chromosome. We'll do 1 in blue, that's version 1, and 1 in green, that's version 2. And then we'll do a little chromosome. So that's like chromosome 1, 2 versions of it. And then we'll do a little chromosome in purple and in orange. Okay, so we're going to look at 2 chromosomes, but we obviously have 23. But we're just going to follow 2. Okay, and right now this is diploid, right? It has 1, 2 copies of everything. So this is diploid. Okay. We're going to go through S phase though, right? So we're going to make copies of these and make those sister chromatids. I'm just going to actually copy that. This is S phase, which is synthesis phase. So what we do, oops. is we make copies of those, right? Boom, just like that. Just like I did. I just made copies of them. Crazy, right? Boom. And they're going to be connected by a little thing called a centromere, right? Like so. I'm not going to draw that every time, but you get it. This is still diploid, right? Because it has two different versions of every chromosome, but we've copied it. And these little things are what? I wish I could. I'd really like to review this stuff with you guys, but these are sister chromatids. I wish. Sometimes the hybrid class, man, I'm like, man, I can't check for understanding. I don't know if any of this is making sense for you. You're probably just slamming through this at 1x speed. Now, if I took like this size sound at normal speed. Okay. Watch this at normal speed. You'll understand it better. Okay, meiosis 1. We're going to transition into meiosis 1. I'm not going to talk about G2 phase. We've already talked about that. It's pretty much the same. And again, right up there, it already says it, but I'm going to put it again. All the interesting stuff happens in meiosis 1. And what's the first phase of meiosis 1? Well, we go into prophase. I'm not going to split up prophase into the different phases. We're just going to talk about just prophase 1 in general. Okay. And let me redraw these cells. Got our nucleus. And what happens is our little chromosomes, let's redraw them. We got our blue. Let me make them a little thicker, actually. Got our big blue. We got our big green, and they actually kind of come together. They get cuddly, okay? And then we got our purple. This is an interesting thing. This doesn't normally happen. We didn't talk about this before. We got our orange. So they're cuddling. The chromosomes are cuddling. What? The chromosomes are cuddling. That's so weird. And it's really not just the chromosomes. Do you remember what these are called? The two pairs of these chromosomes? These are homologous pairs are cuddling. Homologous pairs are cuddling. Okay? And this is prophase 1. Putting that in the color because it's important. Prophase 1. Something really interesting happens here. And I'm going to do a little zoom in of this. So we'll do a little zoom in. Zoom in. Okay? Shoo. We're going to look at these chromosomes. So let's redraw our blue guy. And we'll draw them nice and big. Okay, and we got our green. And the green is actually kind of wrapped around the blue, like so. All right. Sorry, somebody's calling me. Okay, and what actually happens here is frickin' bonkers. So these then will separate like so. But up here, what actually happens is part of the green chromosome is swapped now onto the blue, and vice versa over here. So we got the green and the green, and we got the blue, like that. Okay, let's do this again. This also happens for, this will happen for every chromosome. So we're gonna have our short purple mixing with our orange. Again, the orange is going to wrap around. And it could do it in a different spot down at the bottom, let's say, maybe. But we'll just do them both the same. And we zoom over here. Not really zoom, but continue the saga. And our short purple now has a bit of orange. And over here, our orange now has a bit of purple. Okay, this process is called crossing over. Crossing over, which is the process of exchanging parts of the homologous pairs, parts of the chromosome. Okay, and this is, shrink this down just a little bit over here. This is a huge, this is a major source of genetic diversity. For example, you could have, if this is happening randomly, different parts. So now it's like the green genes are now part of the blue chromosome. And so we're mixing the alleles, like dominant, recessive, so on and so forth. I know we haven't really talked about that in this class, but this is just like increasing our ability to mix up our genes. So for example, if you have a lot of children, none of them will be quite the exact same. They won't be exact copies because the father is doing this and the mother is doing it. Okay, and this is only one source. There's another major source of genetic diversity, or another major source of increasing our genetic diversity. Okay, this little part here is called a synapse or chiasma. Chiasma. Okay, where they come together. Really know this, crossing over, and that this is a major source of genetic diversity, and it occurs in prophase 1. Okay, so in meiosis 1, in prophase 1. Okay, let's continue this on. What comes after prophase? Well, metaphase, obviously. Again, I'm going to put this in a color. Metaphase, because it's also a very important part. Metaphase occurs. And this is metaphase 1, sorry. Metaphase 1. And let's do this too. Let's put some little fire around this because it's so important. Little explosions. Okay. All right, same here. This metaphase, super important. Okay. We know what happens in metaphase. Oops, don't want that to be purple. Nope, there's no nucleus in metaphase. Nope, don't want that. No metaphase. No nucleus in metaphase. This is bothering me. Okay, so our chromosomes are going to line up, and they're going to look like this, right? We've done this little crossing over thing. So we need to represent that. So we've got a blue. I'm going to draw them a little thicker. A blue with a little bit of green at the top. And they line up next to each other. The homologous pairs line up next to each other. We've got a green with a little bit of blue at the top. If you can't represent that, that's okay. The colors, you can do it in a different way. And then we've got a purple, a little bit of orange at the top. And this is the shorter one. And then we got an orange with a little bit of purple at the top. Okay, and we still have our, whoops. And these are actually attached to each other. So they're really closely linked, and there's actually like a bunch of proteins that are holding these together. We don't need to draw those all in. But just know that they are. Okay, then we still have our centrosomes. Oh, way too fat. And we have our microtubules. Okay, this is also really different than before. And I'll go back to it so you can see. We're still going to have our microtubules at the equator and our microtubules kind of overlapping in the center. Okay, so if we go back to metaphase of mitosis, let's go look at it. Right here. The chromosomes lined up like this. The sister, like these are homologous pairs, but they didn't stack up next to each other. They stacked up like that. Okay, so that's way different. Homologous pairs line up in meiosis 1. Okay, metaphase 1. Homologous, and this is going to split those two versions of chromosome 1 or chromosome 2. And this is, now we're going to be haploid after the end of this. Homologous pairs line up in the middle. Okay, and something crazy can happen here. I'm not going to draw this out. Just to save kind of time. I'm going to show this little image here, just because it takes a lot of time to draw those different things. A whole nother source of genetic diversity can occur, okay, in this metaphase 1. So another source of genetic diversity. Diversity occurs. Okay, so the orientation, so these are we'll walk through this. These are different versions that could occur. So this would be like version 1, version 2, 3, 4, 5, 6, 7, 8. Okay? So we have the two different versions of chromosome 1 here lined up like this. But they could be like that. They could be flipped, or these aren't flipped. This is flipped, where the red is now on this side and the blue is on this side, right? So you could see all the different variations of the way this could occur. They could be completely flipped like there, partially flipped here, partially flipped here, partially flipped here, so on and so forth. So you have 8 different combinations, I guess, maybe more, if you have 3 chromosomes. I don't know if any of you have taken statistics or know anything about probability. It'd be interesting to kind of have a statistician tell us. I don't know how to do that probability math. I forgot all that stuff. How, if you have 23 chromosomes, it's thousands at least of different variations this could be. It's probably closer to 1 million or something. That chances of like getting the same combination for a gamete. This is called independent assortment. So it's the independent assortment is the orientation, so the random orientation of alignment orientation of homologous pairs. I think alignment makes more sense. Alignment of homologous pairs. I have such a hard time with that word, just actually typing it. Homologous. Okay, this is super important, right? Because this is increasing, it's just increasing the odds that we're not going to have the same genes coming out in the same offspring. We're going to increase our genetic diversity in our gene pool, which increases our chance for survival. It's pretty crazy, dude. It's really crazy. Okay. Then what happens? We go through anaphase. I'm actually just going to skip. I'm going to skip anaphase because I don't need to really draw it out. Right? These two are going to go over here. These two are going to go over here. So we'll just draw these daughter cells. All right. We'll just draw these daughter cells. And we'll draw them up and down just so we can actually kind of continue their saga. Okay, and I need the colors over here. So we've not actually dissolved the centromere, which is pretty interesting. What we've done, let me just recopy these so I don't have to redraw them. Speed this up for you guys. So this one, these two are going to be together. So that guy's going to go there. And this guy's going to follow it. Whoop. And then these two are going to go down here. Boom. Okay. Right? Because the green, big green is going to be with the orange. Boom. Because these two are going to go this direction. These two are going to go this direction. Okay? So this is the end of meiosis 1. So this would be anaphase, telophase, cytokinesis occurs, all that. And this is the end of meiosis 1. And now we've done essentially 2 methods of increasing genetic diversity, and these are now also haploid, because they only have one version of the big chromosome and one version of the little chromosome. So these are now haploid. Okay? Put some fire around that. That's the third thing that's very interesting. Right? Here we got this crossing over. We got homologous pairs lined up in the middle. Actually, that's the fourth, because that's a really interesting thing. There's 4 interesting things that happen. We got crossing over, homologous pairs lined up in the middle, random assortment, independent assortment, and then the cells are now haploid. Okay, now let's go through just very simply. We'll just show these in metaphase. We'll go into meiosis 2. We're going to go into meiosis 2. With both of these, we'll show them in metaphase. Metaphase, these chromosomes are lined up in the middle. We copy these bad boys over. Oh no, that's not what I wanted. Making my life hard here. Okay, like that. And grab this boy like that. Okay, now these guys come too for the bottom one. Boom. And now you see, now we have our sister chromatids lined up in the middle. Our sister chromatids are going to be separated here. Right? So we'll draw in our centrosomes. Our microtubule organization centers. And then we will draw in our microtubules. Like so. That one looks a little goofy. Oh boy. All right, I need to focus. There we go. Make him a little prettier. Like so. Yeah. Beautiful. And we got our overlappings in the middle. Okay, up here, same kind of story. Microtubules go to the equator. There's actually machinery that all walks along this to signal, hey, this is where we're going to make the cleavage furrow. And I studied a protein that's like a little Swiss Army knife that accumulates right here and helps kind of bring all these partners in together, and it's like a little tension sensor, make sure everything goes smoothly. Okay, again, I'm not going to show anaphase, but what's going to happen is the centrosome that holds these two guys together dissolves. Okay, so this is metaphase 2. And this is just like mitosis. Metaphase 2, right? Just like mitosis. Let's go back and look at it. Right? Just like that. We got the chromosomes lined up in the middle. It's going to look like that. They're going to split off. The sister chromatids are going to separate, right? Sister chromatids line up in the middle. Okay? And then we go through anaphase, telophase cytokinesis. In anaphase, I'm not going to draw it out, but anaphase separates the sister chromatids. Okay, and then they also still go through telophase and cytokinesis. Kinesis. Okay. Guess I'll do it like that. Make these arrows longer. Okay, so now we're going to have 4 cells. This one's going to give us 2. All right, and let me kind of keep them over there in view so I can actually see what I'm drawing here. And this one's going to give us 2. Okay, so this one's going to have these 2. Boom, boom. Blue and purple only. I'll just redraw them actually. So it's going to have big blue, little purple. Right? It's going to have that and that. This one's going to have blue with green. Blue with green. And it's going to have purple with orange. Little purple with orange. All right, and this one's going to have big green, little blue on it. Big green with a little blue on it. And it's going to have orange with a little bit of purple on it. Short orange, a little bit of purple. And then this one's going to have just big green. Yeah, big green, little orange. Okay? Big green, little orange. This is, you can already see like this. And this is all, I'm only showing you 2 chromosomes. So each one of these is a gamete. So now we have our gametes, which are still haploid. Still haploid. Okay, these are our gametes, which are still haploid. And you can see that each gamete has a unique genetic combination of these chromosomes. This gamete has big blue, little purple. This gamete has a mixture of big blue, green, and a mixture of little purple, little orange. This gamete has a mixture of big green with a little blue on it. This one has a mixture of the small. This one is the normal big green, and this one has the normal little orange. So we got these gametes with these norms and these gametes with these mixtures, and that could have been different. Every one could have had a mixture because of independent assortment. You see how kind of crazy all this is. And that's how we make our sperm and eggs. So I already kind of talked about that sperm development and egg development. If we go back to that, let me see, do I have? Thought I had another board kind of pulled up here. Yeah, this one. I can just copy this stuff over. Okay, let's kind of talk about what this actually means. So with our sperm development, it's like such. We have this kind of stem cell here in the seminiferous tubule, and it goes through mitosis actually first to make a copy of itself. And then that diploid cell goes through meiosis 1 to make haploid cells, and then it makes these 4 gametes. And that's how it works with sperm. When it comes to making eggs, it's actually really kind of crazy how this works. So what the egg actually does, this is the egg here, the primary oocyte, it's diploid here, and what it does is it just throws away that other little version of itself. And it doesn't ever do anything. So now it's haploid, and it's only got this blue and this, and there could crossing over and all that stuff can occur. So now it's haploid, it's just got these versions. Okay, and then that's a secondary oocyte. And then it doesn't actually go through that third, or that meiosis 2. It doesn't go through meiosis 2 until it's fertilized by the sperm, interestingly. And it ejects another, what's called a polar body. It's like a small little nucleus that contains that genetic information. So it'll get rid of this version and this version, these sister chromatids. And it does that after it's fertilized. And then it'll become diploid. This will be our zygote. Pretty crazy stuff. Okay, so eggs are really expensive. We don't make 4 of them. Our oocytes are expensive. And this actually, since this is an egg, as soon as it becomes fertilized, then it goes through that process. Yeah, technically now that this has been kind of fertilized, it is a zygote. I drew that a little weird, but that's okay. All right, that's how we make our gametes. Again, meiosis is only to make gametes. We now have haploid. You have all the interesting stuff happening in meiosis 1. That is it about cellular reproduction. Next week, we're talking about cancer, stem cells. It's a really cool topic. Okay, I'll see y'all later.